So much for the invitation. Thanks so much for the invitation to speak here today. Um, I'll be speaking about an overview of iron ferroptosis and Alzheimer's disease and bring you up to date with our work in this area. And there are my disclosures at the bottom of the slide. Well, we are living through an interesting research era in Alzheimer's disease research with a litany of failed phase 3 clinical trials against the dominant target, the amyloid accumulation of Alzheimer's disease. And it's been very challenging to try to cause disease modification with this anti-amyloid approach. And only recently has there been FDA approval of one drug which has a debatable benefit in terms of clinical outcome. And rather than critique it here, I'd invite you to have a look at a recent review that Scott Aiton and I have published in Aging Research Reviews, which goes over the issues with beta amyloid being the sole culprit in terms of the pathogenesis of Alzheimer's disease. And the field now, we believe, is opening up to other possibilities, and it's one of these possibilities that I'll be discussing today. So really, philosophically, Alzheimer's disease has been forced to go back to basics after a focus on beta amyloid and proteinopathy in the last 20 or so years. And what we've been highlighting in our approach is not so much the proteinopathy, which is really conspicuous and a legacy of the neuropathologists upon which the field was founded decades ago, but more the conspicuous death of neurons that occur in Alzheimer's disease. And this figure shows you the really obvious atrophy that occurs within the brain in Alzheimer's disease. Of course, this audience would be familiar with this, but philosophically, it's mysterious that this death of neurons is not given more attention, because even up until now, there's no consensus as to what is the actual mechanism for the neurodegeneration and the loss of neurons in Alzheimer's disease. I mean, it's assumed, it has been philosophically assumed to be related to the proteinopathy, the amyloid and the tangles, but the actual machinery that causes the cell death has been ignored. And I'm going to be showing you the evidence for how ferroptosis, a relatively recently conceived form of regulated cell death, is actually a very strong candidate for causing the neurodegeneration and the loss of neurons in Alzheimer's disease. So our journey towards iron came on the back of the reports of iron concentrations within the proteinopathy. And this slide illustrates the concentration of iron within the amyloid itself, amyloid plaques, and within the neurofibrillary tangles. So these reports have been building up ever since the mid-90s. The report on the right from George Perry's lab with Mark Smith was from 1997. And on the right is a more modern report using more modern techniques in the amyloid plaque on the left, sorry. But there are numerous instances of iron enriching within the proteinopathy of Alzheimer's disease. And in the background of the tissue, there's a great deal of lipid peroxidation. And lipid peroxidation, as I will explain, is the biochemical signature of ferroptosis. But it's interesting, as we studied this field more over the last few years, we became aware that actually the accumulation of iron in the pathology of Alzheimer's disease was commented upon really early in the literature. So more than 60 years ago, there was a report from Goodman that says that the most significant finding perhaps was the presence of large amounts of an iron-staining substance in cells which had undergone neurofibrillary degeneration within the plaques and in the oligodendroglia and in the microglia, in a description of the neuropathology of Alzheimer's disease. And then in 1960, Holmgren and Surand are commenting the occurrence of histologically demonstrable iron in the frontal and temporal cortex is a very characteristic feature in Alzheimer's disease. So iron pathology is in fact a conspicuous but overlooked feature of Alzheimer's disease post-mortem tissue. And if we fast forward to more modern neuropathology reports, here's illustrations from a couple of papers from a Dutch research group, which nicely illustrates the iron pathology of Alzheimer's disease. And here you see iron staining of cortical rind in cases of Alzheimer's disease. And when you have a look at the cells, both neurons and glia are affected by iron accumulation. So this is actually a fairly conspicuous lesion which hasn't really attracted as much attention from the neuropathologists as we may have imagined, the proteinopathy being more dominant, but certainly it is a conspicuous neuropathology worthy of comment. Now, we've tackled the issue of iron in Alzheimer's disease in large cohorts of postmortem tissue acquired from the Longitudinal Memory and Aging Project in collaboration with Julie Schneider and Martha Claire Morris from the Rush University Medical Center. And our approach has been to take the postmortem tissue in their cohorts, which is accompanied with a decade or more of antecedent cognitive performance data, and grind up the tissue and extract the metals and measure the iron directly using inductively coupled plasma mass spectrometry. And in studies like this, we've got large cohorts of tissue, and this enables us to make quite definitive comments about the contribution of the neurochemistry of iron to the antecedent rate of cognitive decline. And I'm just going to highlight the actual results for the patients who, when they died with a diagnosis of Alzheimer's disease, which was confirmed pathologically, for which we have up to 10 years of antecedent cognitive data. And to present these data from these papers which we've recently published, I'm just firstly going to illustrate the impact of the burden of neurofibrillary tangles in the postmortem tissue and how that relates to the rate of antecedent cognitive decline. So in these data, we see on the x-axis the years prior to death, and on the y-axis the change in global cognition. And of course we've got different components of the cognitive performance, but I'm just showing you the global cognition here. And the burden of the tangles in the tissue at postmortem was ranked in tertiles, so low, medium, high, just for illustration, but the results also apply to a continuous analysis. And this shows that, as expected, the neurofibrillary tangle burden is concordant with the rate of antecedent cognitive decline. Now, all of these subjects were cognitively normal when they entered into the study, so all of these subjects converted to Alzheimer's disease during the 10 years of the study. And it is already known that neurofibrillary tangles are associated with cognitive deterioration. So this is corroborated by these data. Next, we look at the iron, the value of the iron in the tissue, and the data here are adjusted for the presence of plaques and tangles and other co-pathologies. And we see, intriguingly, that there is the same type of relationship, that the higher the iron burden, the greater the rate of antecedent cognitive decline in this cohort. And this is, because it's adjusted for the presence of tangles, this shows that iron is an independent pathology that has an association with the rate of cognitive decline. Intriguingly, if you look at the plaque burden in the tissue at postmortem, it has no relationship with antecedent cognitive decline when adjusted for tangles and iron burden. So this is one facet of data that supports a role for iron in the adverse outcomes in the natural history of Alzheimer's disease. I'll now show you from other points of view what other tissue analysis of iron can show, but conclusion from this study is that high iron measured in postmortem brain, this is a composite of 4 regions, was associated with faster disease progression in the decade prior to death in 227 Alzheimer cases from this cohort. So looking at CSF ferritin as a biomarker of brain iron burden, these values were obtained from the ADNI cohort, And we've written a number of papers where we've analyzed how higher CSF ferritin is associated with faster disease progression, whether it be cognitive performance, brain atrophy, or FDG PET. And this is in up to 5 years of prospective data from mapping cognitive decline. In the data that I'm illustrating here, we're looking at the drop in FDG PET, so glucose metabolism impairment, and showing that even when adjusted for the presence of the abnormal tau A-beta 42 ratio, which is now a consensus criteria in terms of the ATN criteria for classifying Alzheimer's disease, that the ferritin levels matter, and that even with the high tau A-beta ratio, we can dichotomize between low ferritin and high ferritin based upon the 50th percentile, in this case, of the cohort, and show that the individuals with a higher ferritin have got a faster cognitive trajectory, consistent with a high burden of iron within the brain, causing a faster, more aggressive disease. Now, we've actually recently elaborated the role of CSF ferritin as a potential predictive biomarker for cognitive decline using findings from the Biofinder cohort. And these are unpublished data in collaboration with Oscar Hansen and Lund. And what I'm going to show you is data from 257 MCI cases with a mean age of 71. And to begin with, I'm going to show you how the classification according to the AT criteria predicts cognitive decline. And we're using on the y-axis, the CDR, some of the boxes, as the performance parameter, and on the x-axis, time measured in months from the sampling. And just on the basis of amyloid and tau values in the CSF alone, you can see that, as predicted, being amyloid positive is associated with a faster rate of cognitive decline. Now, this corroborates facts that are known, that an amyloid positivity in this test in CSF values is associated with cognitive decline. So what we did was then analyze the CSF ferritin as we did previously with the ADNI cohort, adjusting for the presence of the other values. And we searched for the optimum ferritin level in terms of being able to classify decliners from non-decliners, and came up with an optimal amount of 12.5 nanograms per mil. So a value higher than 12.5 nanograms per mil, which is about half a standard deviation above the norm in non-MCI cases, gave us the optimal performance in terms of predicting cognitive deterioration. And so when you have a look at the cognitive decline over the 4 years on the basis of baseline CSF ferritin, dichotomized at that 12.5 nanograms per mil cutoff, you can see those with higher CSF ferritin decline faster than those with lower CSF ferritin, and that the effect size is about the same in terms of performance as the A and the T criteria. So ferritin could be conceived of as an independent biomarker in terms of predicting cognitive decline with a comparable effect size to using amyloid and tau. And yet, of course, it's not meant to be, on the face value, associated with the biology. Just drilling down further into the different AT classification groups, we can see that the strongest impact of this ferritin dichotomization is in the amyloid positive, tau negative group, where having an elevated ferritin level is associated with a very aggressive cognitive decline in these MCI cases. And it would seem that in advanced disease, where the patient has already biochemically Alzheimer's disease because they are A+, T+, that the ferritin has a minimum impact. So we're using this information in order to put together a picture about where the elevated iron in the brain might fit into the natural history of Alzheimer's disease. A final approach in terms of measuring brain iron is using MRI imaging, and particularly a modality which we use in Australia, which is quantitative susceptibility mapping. And we've applied that to our Australian cohorts, the ABLE cohort. And these are data from a paper which we published recently, which showed that high brain iron measured by QSM is associated with accelerated cognitive decline. And this is involving a cohort that has been followed up from baseline every 1.5 years over a period of 6 years. The heat map in the center of the slide shows you the statistical average increase in iron throughout the cortical regions. And on the left, we see a graph of the relationship between baseline iron levels in these cases and the subsequent, on the y-axis, the change in z-score in standard deviations per year for this cohort over the 6 years. Now, in amyloid negative people that have been assessed as being amyloid negative by PET imaging, there's no relationship between the change in z-score, the rate of change in episodic memory over the 6 years of the study, and the burden of iron. However, in amyloid positive individuals, these cases are very sensitive to the burden of iron within the brain. So there's some sort of interaction where iron burden in the brain matters very much once amyloid has started to deposit, and that it is the burden of iron which predicts the cognitive decline in these individuals, these amyloid positive individuals. So putting it together, and this theoretical position is explained in this paper in Alzheimer's and Dementia. Previously, iron had been implicated in the proteinopathy of Alzheimer's disease, perhaps by facilitating the formation of plaque or the formation of neurofibrillary tangles, or making the tangles and plaques more toxic if they are toxic, but the mechanism of neurodegeneration was not clear. What we're now pursuing is, based on the data that I've just shown you, is that actually within a population, the burden of iron within the brain matters. And this burden is probably normally distributed, and it appears to impact the natural history of Alzheimer's disease most strongly at a point after plaque deposition has begun. And I'm going to continue now to explain how we think that the most likely mechanism for this iron to be involved in neurodegeneration is through the cell death mechanism ferroptosis. Before I get into ferroptosis, I want to make a comment about iron in terms of the natural history of Alzheimer's disease and aging. Obviously, the major risk factor for neurodegeneration is aging. And the chemical changes that occur within aging are very important to understand in terms of the risk for neurodegeneration. So could iron be a factor that increases in the brain with aging, and so increases the risk for neurodegeneration? Well, in fact, once again, this has already been commented upon over 60 years ago, where Hallgren and Surander did a survey of the amount of iron within the brains of individuals over the human lifespan. And this is an extraordinary paper which shows for the first time the level of iron within natural aging in the brain. And you can see that at birth, the levels of iron are very low. They rise rapidly throughout childhood, and they plateau in adulthood, but they still drift upwards as you age. This trend has been noticed also more recently in applying modern imaging techniques such as QSM and relaxometry. And this has confirmed the age-dependent elevation of iron within the brain. Now, obviously, the brain uses iron for metabolic purposes, particularly in the formation of heme, so that the growing brain needs a supply of iron for neurodevelopment, and iron deficiency is hazardous. But after development has been completed, say from the age of 25 onwards, why is there this continued elevation of iron? Conveniently, it can be studied in animal models. And this is data from our first paper on the subject, which measured the amount of iron in the brains of mice as they age. And you see in the graph on the right, the levels of iron at 2.8 months of age, 8 months of age, and 18 months of age in wild-type littermate controls of the APP transgenics. This is the TG2576. And there is this age-dependent iron elevation that occurs after the reproductive life of the animal, which is about 6 months. But interestingly, the APP overexpression seems to oppose this elevation of iron. And in fact, this turns out this was the first clue we had that APP participates in iron homeostasis in the brain. The elevation of iron in mice, in the brains of mice, is a robust and reproducible phenomenon, and we've reported how it's influenced by the sex of the animal and the background genetic strain of the animal. And recently we've reported that APP knockout mice have got an exaggeration of this age-dependent iron elevation in the brain. And I'll go more into the machinery of this as we go along. So the literature filled up with instances that reported this age-dependent iron elevation. And it was reported in rats and other rodents. It was reported in primates. It was reported in the human brain again, and using imaging studies of the human brain became very well consolidated in the literature, with numerous reports, again reporting that brain iron deposition occurs over the adult lifespan, and that this has been elaborated with very sophisticated studies, and the imaging of brain iron is now quite a common report in the literature. And the consensus is very clear that it rises post-adulthood for mysterious reasons and is associated with cognitive loss and disease pathology. And I'll tell you more about this as we go along. Intriguingly, the iron accumulation doesn't just affect the brain. It also affects skeletal muscle. And this is a report that we had where we surveyed the amount of iron in different organs. The tibialis anterior, comparing adult mice to aged mice, and the elevation of iron was also seen in the liver, in the fat, in the kidney, but not so much in the heart. So in addition to the brain, there are other organs that accumulate iron, again mysteriously. It's not clear why this happens. We use laser ablation ICP mass spec to map the iron elevation within the tibialis anterior of the mice, and at the same time we also showed that the tibialis anterior tissue has got elevated 4-hydroxynonenal levels, and 4-hydroxynonenal is a reactive aldehyde which is a product of lipid peroxidation, which itself is a product of abnormal iron combustion with polyunsaturated fatty acids, and I'm going to tell you more about this as we go along. So iron can be toxic by several means. Excess iron can form the hydroxyl radical, which is damaging wherever it is generated. Also, excess iron can suppress antioxidant transcription factors. But where I'm heading is to talk about ferroptosis. And this is an iron-dependent form of non-apoptotic cell death, which is described as a regulated cell death mechanism in the defense against cancer and infection. And it has been increasingly described of interest in the literature since the first report in 2012. And classically, what ferroptosis generates is lipid peroxides and uses as substrates long-chain fatty acids such as arachidonic acid, which you see in an oxidized form surrounding plaques. And here's a nice illustration of it by Paul Axelson's group. So what is ferroptosis? Ferroptosis is a reaction where the ferrous iron, which is present at about 2 micromolar in the cytoplasm, reacts with polyunsaturated fatty acids, becomes oxidized, and generates lipid peroxides. And the lipid peroxides can be lethal by causing plasma membrane disintegration and rupture. This reaction can be blocked by small molecules, and a potent radical trapping agent is liproxtatin, which I'm going to show you in experiments in a minute. But also vitamin E acts at this point. But vitamin E is a very, very weak version of liproxatin. Whereas liproxatin is effective with an IC50 in the low nanomolar range, vitamin E requires nearly millimolar concentrations in order to achieve the same result. And of course, trapping the iron itself with chelators, such as deferriprone, which is an orally bioavailable clinical iron chelator, which is in clinical trials, is one way of lowering iron. But I'm going to show you some data using SIH, which is a convenient iron chelator that I'll show you in experiments in nematodes. At the same time, the lipid peroxides, when they fragment, generate reactive aldehydes, such as 4-hydroxynonenal and malondialdehyde. And it's already been reported that these reactive aldehydes can induce the proteinopathy, which is classic of Alzheimer's disease and other conditions such as Lewy body disease. And glutathione plays a critical role in this reaction because the checkpoint enzyme that blocks lipid peroxidation from being propagated is glutathione peroxidase 4, which uses glutathione as its substrate. So in getting on to, I want to show you a very interesting set of experiments involving C. elegans. And before that, I'll just say that ferroptosis is universal. Iron accumulation in the kingdoms of life is universal. And here we see the levels of iron accumulating within the bodies of Drosophila as they age. This is a report from 1985. And we took on the challenge of studying this in C. elegans. And first we characterized the same phenomenon that occurs in C. elegans in a couple of papers where we showed that as the animal ages during its adulthood, that there is accumulation of iron within its soma, particularly in its metabolic tissue. And here you see an illustration of iron-stained C. elegans. The animal is dissected into head, middle, and tail, and it's stained for iron. You can see this vivid accumulation of iron between 4 days of age to 12 days of age. The animals live a median lifespan of about 8 days of age, and they're all dead by 20 days of age. So it's a very convenient system for determining whether or not this iron accumulation is adverse in terms of outcomes for longevity and health span. And that's exactly what we did. We manipulated the iron in these animals using small molecules. And so I'm going to show you data from this paper, which we published last year in eLife. And I think it's very interesting data, and want to go through this a little bit carefully. Here, first I'm going to show you the survival curves of these C. elegans. And this shows you standard survival curve of the control group, 709 worms over their lifespan, with a median lifespan, as I said, of about 8 days. These animals were treated from day one with either liproxatin, which is a high-affinity radical trapping agent, which is a classic anti-ferroptosis reagent, or they were treated with SIH, which is an iron chelator that we established can pass into the worm through its gut. And what we see is treatment with either of these agents more or less doubles the lifespan of the worm. So that the median lifespan now is increased by about 100%. And at the same time, we use the synchrotron to map the iron in the worm. We have a synchrotron here in Melbourne. So, we're in lockdown. We get a bit bored, but certainly a synchrotron is a way of amusing yourself. And the synchrotron established that the iron is elevated quite conspicuously as the animal ages. From here we see a comparison of day 1 to day 4 to day 8. And then in the liprox-treated worm, there's no change in iron, which is what we would predict because liproxatin is not an iron chelator. It is a radical trapping agent that stops the consequences of iron. But in the SIH-treated worm, you see this normalization of iron, returning values to pretty close to what they are at day 1, despite this animal being 8 days of age. So we have succeeded in lowering the iron or preventing the iron from causing lipid peroxidation, and as a result, we've caused this doubling of the lifespan of the worm. Here we see a quantification of the iron within the bodies of the animals, showing that lipoxystatin does not affect the iron levels in the animal, but the SIH keeps the iron levels low. So it's interesting that the animal can proceed throughout its adult lifespan and through its reproductive period with maintaining low levels of iron. So this iron accumulation is so unnecessary that the animal can live normally even if it's deprived of iron at this age. And what else is intriguing is that you can see quite obviously that the SIH treated animals with the low iron are actually larger than their control animals that have not been treated with a chelator. The lipoxystatin animals are not changed in size. So we can quantify this, that lowering iron in these worms actually creates a bigger animal, and not only that, it creates a faster animal, as does the liprox. So treatment with either an iron chelator or a radical trapping agent not only increases the lifespan of the worm, but it increases the health span of the worm. And so this is quite a remarkable finding, that we can achieve these effects with no metabolic price paid for the intervention. So these animals have not got a slowed metabolism, such as the other lifespan mutants that you might see in this model. So this allows us to make some big picture statements about this iron accumulation. Life obviously depends upon iron, and evolution depended upon the availability of iron in the Earth's crust. However, because iron is so essential for development and was a limiting factor in terms of biological evolution, there's been no pathway evolved for the excretion of iron. And this is very interesting, that if you consider mammals, the only way that we can lose iron is either through bleeding or through desquamation of the gastroepithelial lining. There's no other way of shedding excess iron. If we ingest iron, if you have a big steak tonight, you will put that iron somewhere in ferritin, somewhere in your body, and it will keep accumulating. And as I've mentioned, obviously too much iron can cause problems. So our hypothesis globally is that ferroptosis, which is normally an anti-cancer defense or an anti-infection defense, eventuates from the needless accumulation of iron with aging. And this activates inappropriately this ancient defense eventually in life and in various diseases. So why is the brain so vulnerable to ferroptosis? So it has the following risk factors. It has high and dynamic iron concentrations. It's post-mitotic tissue. It's got a high oxygen turnover, which enables the lipid peroxidation to proceed. And it's got a very high polyunsaturated fatty acid content. And of course, it has got this obvious age-dependent increase in tissue iron. So this is a perfect storm of factors which renders the brain so vulnerable to ferroptosis in later life. And just surveying the literature, the features of ferroptosis that are seen in the pathology of Alzheimer's disease are quite numerous. And these are all reports that I've just taken out of the literature. But these types of changes are also seen in other diseases, by the way. So maybe this is a generic neurodegenerative mechanism. So importantly, and this is where the research is heading right now, if this is a reasonable account for the actual neurodegenerative mechanism of Alzheimer's disease, how do we then explain the genetics of Alzheimer's disease, which have they've been so emphatically framed towards proteinopathy, so that the mutations of the amyloid protein precursor and presenilins, which I'm sure this audience is familiar with, are said to cause amyloid deposition, and the mutations of tau, which cause familial tauopathies, are said to cause tau aggregation. But is there a way that these mutations could explain vulnerability to ferroptosis or iron metabolism? Well, the short answer is yes. And this is a very long project. And in this talk, I'm not going to be able to touch on all of the features of it, but I just want to update you to where we are. This is a cartoon of iron being effluxed from a cell into the extracellular milieu. And the obligate pathway for iron efflux from cells is through ferroportin. Ferroportin is on the surface of the cell. It connects to ceruloplasmin, which then oxidizes iron, which egresses through the cytoplasm, through the ferroportin, and loads up transferrin for iron to be trafficked elsewhere. Now, Jack Rogers and Rudy Tanzi and myself published quite a while ago that there is an iron-responsive element on the 5' UTR of APP, which is sensitive to the concentration of iron within the cell and generates the production of the amyloid protein precursor. And the APP functions to stabilize surface ferroportin. And tau participates in this function by chaperoning the trafficking of APP towards the cell surface where the APP docks with ferroportin and stabilizes it. And these papers, which I'm illustrating, just show you some of the reports that have described this machinery. So full-length APP stabilizes ferroportin and enables the efflux of iron from the cell, lowering the cytoplasmic content of iron. So this machinery is set up to be able to influence the concentration of iron within the cell. And we showed that tau knockout mice have got an elevation of iron within the cortex, hippocampus, substantia nigra, not the cerebellum, not the liver. And that if you study primary neuronal cultures of APP, of tau knockout mice, that they are slower to efflux and iron burden compared to wild-type primary neurons. And the amyloid precursor protein, I'll show you the data of it being pulled down with ferroportin from mouse brain and human brain tissue. It's pulled down the same way as ceruloplasmin is also pulled down in this complex, which facilitates the efflux of iron. APLP2 was used as a negative control here. That's not complex with the ferroportin in the pull-down assays. And APP knockout neurons in culture are slow at effluxing iron from iron-loaded cells. But this is where I wanted to head to today, and this is the first report of the impact of a genetic mutation of APP on this iron efflux mechanism. And it was published last year in Molecular Psychiatry. And it studies experimentally the impact of either the Italian mutation, which is a mutation of the second residue of the A-beta domain within APP that causes aggressive familial Alzheimer's disease, or a mutation of the same residue to threonine, which is the Icelandic mutation, which is a very unusual mutation, which is actually preventative for Alzheimer's disease. So these experiments are very nicely matched in terms of the site of the mutation. And it's already been well studied that the Italian mutation promotes the generation of beta amyloid through beta cleavage through base 1, and that the Icelandic mutation protects against base cleavage and enables alpha secretase cleavage to proceed. So in this paper, what we did first was to create stable transfected cells with these models. But first we studied the effect of pharmacological inhibition with either TAPI1, which is an alpha secretase inhibitor, or B4, which is a beta secretase inhibitor. And we took N2A cells that were loaded with iron, and here you see the elevation of iron that occurs when you just challenge with 50 micromolar iron. And inhibition of the alpha secretase drives up the iron concentrations further, and inhibition of the beta secretase relatively normalizes the iron. And here you see in the Western blots the impact upon APP beta generation and other things, including ferritin. So we see modulation of the alpha and beta cut does alter the retention of iron, so that it looks like the alpha form of APP facilitates iron efflux, that inhibited by the base inhibitor. At the same time, the base inhibitor causes an increase in surface APP measured by FACS sorting, and this is accompanied by an increase in cell surface ferroportin. So this is the obligate iron efflux pathway. So the stabilization of APP on the cell surface stabilizes the ferroportin, and this is consistent with stabilizing the iron efflux machinery of the cell. Now, doing the same experiment but with RNAi, we could knock out atom 10 or knock down base 1 and achieve the same results. So knocking down the alpha secretase causes a retention of iron, and knocking down the beta secretase causes iron normalization. And again, this is associated with the stabilization of APP, stabilization of ferroportin due to the presence of more APP on the surface of the cell. Now, looking at the mutants themselves stably transfected into the culture, we take these cells and iron load them, and we can rescue the iron load, as I mentioned, with a beta secretase inhibitor. So this just shows you the performance of wild-type transfected APP cells compared to the basal levels. So the iron levels go up, but inhibiting the base activity normalizes it. If you look at the Icelandic mutations, they are resistant to iron loading. So the iron levels are less than the wild type APP, and base doesn't have much of an effect there because it's already relatively unperturbed. But if you look at the Icelandic mutant with the representing aggressive familial Alzheimer's disease, you can see that the iron burden is much greater in these cells and normalized by treatment with the base inhibitor. And this comports with the presence of the APP on the surface. So the Italian APP is found in less abundance on the surface of the cell compared to the wild-type APP, and the Icelandic APP is more abundant on the surface of the cell. And this correlates with the presence of the ferroportin. So there's less surface ferroportin in Icelandic APP, and there's more surface ferroportin. Less with the Italian APP, there's less surface ferroportin, and there's more surface ferroportin with the Icelandic APP. So this is the first genetic report of the genetic lesion of Alzheimer's disease influencing iron efflux. And just to summarize what I've said is that APP functions to stabilize ferroportin, but it does so if it's either full-length APP or if it's cleaved at the alpha cut. So SAPP alpha appears to be able to stabilize surface ferroportin and facilitate the egress of iron through this channel to load up transferrin. But if the APP is driven through the beta secretase pathway, SAPP beta is unable to stabilize surface ferroportin, which then gets degraded within the cell and prevents the efflux of iron, causing iron accumulation within the cell. So this is what we think is the first evidence of a pathogenic mutation of Alzheimer's, of APP, influencing iron accumulation. And basically what this also tells us is that the generation of beta amyloid is a signature of iron accumulation within the cell, that they happen simultaneously but are not directly associated, because the same machinery that generates beta amyloid is also responsible for the accumulation of iron within the cell. So to wrap up, I'm just going to have a quick look at the clinical developments in this field. I wanted to mention that Parkinson's disease is a related disorder of iron accumulation. It's a different part of the brain which is affected, and that's the substantia nigra. And here you see some relaxometry images of the iron accumulation within the substantia nigra, a report by David de Vos in Lille, and he quantified this accumulation of iron in these regions associated with severity. And the accumulation of iron within the neocortex in Parkinson's disease is associated with the cognitive loss, which is a problem in Parkinson's disease. It causes a form of dementia as well. So it's already been noted that this iron accumulation appears to be sinister in a manner that's similar to Alzheimer's disease. So there have been a couple of shots on goal with these targets. Firstly, in 1991, Crapper McLaughlin and colleagues published a paper on intramuscular dose ferrioxamine in a phase 2 clinical trial of patients with Alzheimer's disease. and reported benefits after 6 months treatment. But this result was not ever attempted to be replicated and was left there in a sort of orphaned report. In 2014, a significant result was achieved in the treatment of Alzheimer's disease in a phase 3 clinical trial using vitamin E. Now, vitamin E is a weak anti-ferroptosis agent, and in fact they used very large doses in this trial, 2,000 units a day, which is about 5 tablets, supplement tablets a day. And this report describes one of the few phase 3 clinical trials which shows a successful intervention. But it's not really commonly used in practice, but it is out there as a successful phase 3 clinical trial. David de Vos's group then reported in 2014 on the use of deferoprone, which is an orally bioavailable iron chelator which is used for the treatment of pediatric iron overload disorders associated with transfusion. Subsequently, they went on to test this compound in Parkinson's disease and reported successful results in phase 2 clinical trials, which were replicated subsequently in a group from London. And this has now gone on to complete a phase 3 clinical trial, and the last patient out has been recently, so we'll know the results of the phase 3 clinical trial for Parkinson's disease hopefully this year. In Melbourne, we've been studying deferoprone to treat Alzheimer's disease. This is a protocol where we're doing a phase 2 study. So it's a randomized clinical trial, placebo-controlled, and we're hoping to have results within the next 2 years. So thank you for your attention. Here is the group in Melbourne who has done a lot of the work that I presented there, particularly Scott Ayton, Abdel Baladi, Adam Southern, and Mark Greeno's work on cell culture. I've mentioned our collaborators, the Rush University Medical Center, Julie Schneider, the late Martha Claire Morris, who was a delight to work with, and from Lund University Medical Center, Oscar Hansen and his team. And of course, I've had a happy collaboration with the Mass General for many years, since I am an alumnus, and that's with Jack Rogers, Rudy Tanzi, and the late Rob Moyer. Well, thank you very much. Here are my credits for the funding, and I'm happy if we have time to answer a couple questions. Thank you.